Hello, my name is Serghei Mangul. I'm excited to present our work about robustness and reproducibility of computational genomics tools. That's a great honor for me. Usually I used to come to CAMDA as a trainee, but that's my first talk as the PI, so I'm really excited about that. To say that all the work was done by Pilin, a talented graduate student at the time at Georgia State, but now she's the postdoc in Zurich. Okay, great. So a couple of definitions before we kind of move on. So first, reproducibility. So by reproducibility, we mean the ability to produce identical results across different runs, and the runs can be done across different laboratories. So for example, you would take a sample and you would sequence that in California, in Los Angeles, and New York, and you want to get the same results. So robustness, we mean to conserve the accuracy of the bioinformatics tools across the altered data, which is a result of different runs. So access reproducibility is really the challenge, and the challenge is it's costly. You need to basically double the cost and shipment, and sometimes it's just impossible for the very, very valuable clinical samples. So how we can access reproducibility is one is, again, so real technical replicates are costly and time consuming. So what can we do? So actually we come up with the solution. So we propose to use computational replicates, so which can be alternative way to real replicates. They will be computationally generated, and they can assess lower bound of reproducibility. So meaning that they cannot capture the entire variability due to different runs or sequencing your sample in different laboratories. However, they can capture some properties. So for that, we have developed CompRep, computational reproducibility, a tool to generate replicates. So so far it has two very simple operations. So operation number one, which you can see on the left, which we call technical replicates type 1, which means just to resort the data. And what you can see on the right, that's the technical replicate number 2, and that is to take reverse complement of each read. So having those two operations, we want to ask the questions if the bioinformatics tools will maintain the same accuracy or the same results as we alter the data. So in terms of the data sets, we have randomly selected 10 samples of DNA-seq and RNA-seq files. And for the alignment purposes, we have considered the reads where you have only the primary alignment being reported. So we have taken few tools in terms of read alignment, several tools for the structural variant detection, and for gene expression, we took Calista and Selman. So as we try to evaluate the consistency, we look at a couple of things. So we look at the reads which are identical, obviously, by identical we mean they map to the same position, and then the way they're mapped is the same. Then we talk about the consistent global, and this is what I'm talking, it's on the left here. So consistent global and inconsistent local, meaning the global position is actually the same, however the local alignment is different, and then we measure the local alignment and looking at the eddy distance. Then we have inconsistent global, meaning the reads are different, mapped to the different positions and as well as different eddy distance. And multi-mapped reads, meaning that in the original data, the read is mapped to the position, let's say X, but in the altered data, the read is mapped to the position Y. However, the eddy distance is the same. So really, the read is multi-mapped, but we just didn't know that. So the tool which classified the read as uniquely mapped reads was wrong in that sense. And then we have type inconsistent 1 and inconsistent type 2, meaning that the read actually was reported with the primary alignment in the original data, but was not reported or reported as not a primary alignment in the altered data. And the results are on the right. So first observation that you can see that doing reverse complement is quite challenging for the tools to maintain the same results. However, resorting doesn't affect all the tools. So many tools actually are exactly the same. And actually, I'm not sure if you will see it. I mean, maybe you will see my picture or like my video instead, but what you can see, the subread, snap, and small, they actually, all the lines go all the way blue, meaning that they maintain exactly the same. There is no effect of resorting. However, you can see every single tool is affected when you do reverse complement, just because it's more challenging. And you can see that the most effective tools are Bowtie and Minimap2, but they are very different in terms of compositions. They're very different in terms of how they're affected. So we also looked at the structural variance tool. So we took the output of read alignment tool and provide them for free structural variant detection tools, meaning breakdancer, DALI, and pop-indel. So what you can see, there is quite a big variability. So first of all, in terms of how read alignment affects structural variance result, but also in terms of how the results are impacted by the technical replicates type 1. So what you can see, the most effective tools was pop-indel, and then the tool was affected quite a bit. And the reason is we actually follow up with the developers of the tools, and we realized that the way they call the structural variants, they have certain thresholds being implied, where if you observe certain number of reads which you're happy with, you would infer the boundaries, in this case deletion, and you will not examine other reads. So meaning that resorting the reads can change that, right? So it happened to be that on the top of your list will have certain reads which you will infer the boundaries in one way, but if you have different reads, you will insert boundaries in different way. And the reason is just because you don't examine all the reads. So I think that's very important, that you do this trade-off, which you're not necessarily aware of as the user of that tool. So I think that actually the great illustration of our metric, which we hope will be adopted, that is really important to challenge the bioinformatics tools, and this is and then the trade-offs between the accuracy and the speed. And then just to clarify what you see on the left, that's threshold 0, meaning that we require the identical boundaries of the deletion in the original data and the altered data, and then the threshold 1000, we allow that 1000 differences between the boundaries in the original data versus the boundaries in the altered data. Okay, so what we did last is that we have looked also the RNA-seq, and that's just like the start of our work. So we have done so far Selman and Callista, and then we did exactly the same thing. We took computational replicates type 1 and type 2, and we tried to challenge the tools. So what you can see, there is some differences, maybe not that dramatic, I would say, between Callista and Selman, but there are some. And meaning that for the Callista, you can see that, for the Selman, you can see that mostly the genes, low expressed genes, or in this case actually transcripts, affected. However, for Callista, you can see even like the highly expressed transcripts are affected. So we're going to explore it more and ask the question, is it only affecting the transcripts? Does it affect the gene level? And so on. But there is something here. And I think another important conclusion is that you resort the data, it does not affect the tools at all, rather only the computational replicate type 2 effects, meaning then you have reverse complement. So I think just to stay on time, even though it's recording, so I don't know actually how it's enforced, the timing, but just to conclude and be on time, that so we have introduced the new tool, CompRep. It's enable cost-effective assessment of reproducibility of genomics tools. We observe substantial variability across the genomic tools, including read alignment, structural variants, and RNA-seq tools. And then we hope this study will inform broad audience in terms of reproducibility, and we hope that's, given how accessible this metric is, it doesn't require any cost, we hope that will be adopted in addition to this kind of standard measures like accuracy and precision, sensitivity. So we hope that will be adopted for the newly developed tools, and actually in the benchmarking as well. Okay, so that was really a teamwork, so many people being involved, so primarily Pilin, who is the postdoc again at the University of Zurich, and Alex Zelkovsky, my PI from my former PhD advisor from Georgia State, and other people. So, and just to conclude, so if you want to join the Mangul Lab, so it's actually actively growing and hiring. So if you're let's say if you finish a PhD looking for the postdoc in sunny California, enjoying the views as you can see on the picture, please email me, and please join the lab. So we're gonna be excited to work with you. Okay, thank you so much. So I think that's all.